All right, welcome to second day of the symposium. It is my pleasure to introduce Dr. Laxmi Parida. So she has made outstanding contributions in the areas of computational biology and bioinformatics. At IBM Watson Research Center US, she spearheads research initiatives in the areas of quantum computing, computational genomics, cancer genomics, topological data analysis, and pattern discovery. I also congratulate her for being awarded the Rukmini Gopala Krishnachar visiting professorship at IISc. We just heard that last week. And for her work, she has been recognized with IBM fellowship, ISCB fellowship. I look forward to her talk today, and we're happy that you accepted our invitation to fly all the way from US to India. Thank you.
My pleasure. So thank you for the kind introduction, and thanks for inviting me and giving me this opportunity to share some of my work with you here. So I will begin with the history, and so I'll begin with something called, this book called Brilliant Blunders, which came out a few years ago. It's not a new book, and it talks about the blunders that top scientists made. Geniuses, as he calls it, including Darwin. And Charles Darwin needs no introduction to himself or his work. In fact, Professor Majumdar yesterday covered in details, quoted from his books and so on about him. It's his theory of evolution and other things that we talk about. So what was the blunder he made? You can already see a clue there. And the years I'm putting there is their prime work was around the same time in the 19th century, while Charles Darwin was proposing blending heredity, and Mendelian, of course, Mendelian heredity. And what it means is that, roughly speaking, so Charles Darwin's theory at that point, he believed, was if a yellow parent and a pink parent will produce a purple offspring, whereas Mendelian had yellow and pink parent will have some yellow, some pink, and some purple offsprings. So what it means is that Mendelian's heredity already indicated that we had discrete units that we were inheriting from our parents. And now, of course, we know it's DNA and so on. So that discrete nature is what kind of drove a lot of computer scientists maybe 100 years later into this field to contribute in the general field of biology. But genome is the blueprint in biology, so everything begins there, I would think. So I'm now going to fast forward, skip one whole century, and come to the turn of this century. And the beginning of this century, we had this data explosion. Data explosion in terms of course, now we think data in terms of text, images in general, but there was this genomic explosion, genomic data explosion that happened at the turn of the century with the completion of the Human Genome Project. And so for biologists here, for the folks here, I think that genomic explosion was very important. People worked in that, and this field of bioinformatics or using algorithms in this space was more or less born out of that. And that's when it came about. Because even late last century, I don't think there was a subject called bioinformatics. And people were debating, will it even be a subject or not? And I've heard some jokes about it, but I don't have time for the jokes, but I'll just sort of move on. And the next one, maybe a decade later or so, is this compute explosion. And when I'm saying compute explosion in terms of computing processing, computing processors, AI, and so on, and now ChatGPT and other things, AI has taken over the world. So these were the two sort of explosions that we are seeing at the turn of this century. And people often ask me, because my background is in computer science, a little bit of math, but more of computer science, but I try to work on problems from biology. And the question that they ask me is that, what are you guys doing that moves the needle for biologists? What are the problems are you solving for biologists? Of course, we are busy solving a lot of problems, but what is moving the needle for biologists? And I think about it often, and I have an answer which has I have some answer which has consistent for the last few years. It'll change, it'll get stronger, and so on. So I just wanted to kind of share that part. So I think the genomic revolution that happened, the genomic data at least revolution that happened, of course, at that point when the human genome got sequenced and so on, we did think that we will begin to understand living organisms at a very fundamental level from the blueprint. But of course, it has been harder, it's taking time, and we are still learning. But the fact that that explosion happened, and now in biology, cataloging biological organisms is actually first, you start with the genome sequence, not first, but you do want to have the genome sequences in, and this is a next generation sequencing and so on. That happened, of course, due to improvements in biotechnology, but also equally due to algorithms and bioinformatics. If you didn't have algorithms for sequencing and for assembly and so on, this would have never happened. So I think, yes, we moved the needle. Algorithmicists or computational scientists in that sense for biology, we moved the needle on that. And then I also bring CRISPR in that category, and I'll tell you why. And CRISPR actually, Emmanuel Charpentier and Jennifer Doudna got the Nobel Prize in Chemistry for understanding this mechanism of acquired resistance in bacteria. When this is acquired because this is the viral attacks that they understand, and they pass on the information to the progeny, the memory that they have. And this is called CRISPR, and now I think it's kind of common knowledge, with gene editing and so on, CRISPR is being used. And I learned from Jennifer Doudna that this germ of an idea to understand this acquired immune system came from 3 papers that appeared somewhere around 2005, in 2005. And these 3 papers were, I'll go back to this and say what they noticed. They noticed that when they were cataloging these bacterial sequences, that they noticed that they had these repeats, So they had these kind of repeats here, these short repeats that were clustered, and in between these, there were these foreign elements, which were viral segments. The viral segments were these segments here. And how did they notice that? And it was through actually looking at all these sequences, doing all the string algorithms that a lot of people do, looking at patterns and so on. And how do you know it's a foreign DNA element? Of course, DNA, it's just ACGT, so how are they foreign? That you also see through patterns by looking at databases of viral sequences and so on. So it is these algorithms that actually brought this to the forefront. And in addition to that, they always found a Cas gene, or there was the homolog of a gene sitting next to these repeats, which is implicated in DNA repair. And so this is the finding that these 3 papers found by using this kind of pattern discovery and other techniques and so on. And that got the biologists actually thinking that, making the connection that perhaps this has something to do with the immunity, something to do with acquired immunity. And they carried out experiments, confirmed everything, and so on. So there's a lot of work, of course, done by the 2 scientists. But the germ of an idea came from these papers, which were using techniques which would have been using techniques that comes from this community that designs algorithms in this space, string algorithms, patterns, and so on. My early career also, I was doing a lot of this pattern discovery and so on, which Chirag mentioned. I even have a book on that, but that's like a couple of decades old now. So I think that we did move the needle, okay? At least we contributed to moving the needle here quite importantly in terms of the algorithms that we do. And the other one that I think that a lot of these other techniques and so on, we will still need phylogenetic algorithms and other things in this space of retrodiction, which I call retrodiction, meaning given what you have today, you want to know what happened in the past. So this is just the opposite of prediction. But you want to know what has happened, what has led you to come here, and phylogeny and others plays a very important role. I was going to say a little bit about the hominins and so on, but I think Professor Majumdar yesterday covered in great length and very beautifully covered that space, so I won't go into that. But I'm actually showing a recent result that you may have seen. Is anybody familiar with what I'm talking about here? Okay. Do you recognize, these are trees. Do you recognize these trees? Sorry? Yes. Yeah, baobab trees. Maybe exactly. So, so these are baobab trees. You know, baobab trees have these pods. You can make drinks out of it and so on. And you see them in Africa and Madagascar. And you also see them in Australia. Okay. So of course biologists had the question, how did it originate? Was it parallel evolution? How do you have these baobab trees in Africa and in Australia? In fact, I'm told there are some baobab trees in India too. In Mumbai, people go baobab tree hunting, and I have some jokes on that, but you know, it's for another day. And so, and then this new result that that was in the news maybe a couple of weeks ago was that they studied the genomes of these, and they concluded that it originated in Madagascar. So intuitively they were right, because there are about 6 species of this tree in Madagascar. So they had enough time to develop six species, so they probably originated in Madagascar. Now the paper says that's where they originated, and maybe the seeds kind of floated down to Australia, and you see the baobabs there. I thought that I will put it in retrodiction, because you can, biologists and all scientists are always asking the question, how did things originate? What is the explanation for what we see here? And this is also a space where we can actually, contribute well in this space. So now I want to go back to the topic of my talk. When I was sitting there, people are asking, so what are you going to talk about? I said, wait and see, let's see what we're going to talk about. So the technique that I'm going to talk about and what we use in these algorithms, it's different from the usual algorithms or AI and so on, and that's topological data analysis. Okay? If you're already familiar with it, I will ask you to bear with me, but if you're not, I will try to give you a perspective from this, from the viewpoint of why should I care, okay? So given that, I'm going to give you some toy examples, try to walk you through that just so that hopefully you'll appreciate what it does. So I'll begin by showing you a picture here. So imagine you have some data. This is a toy example. So you have data where you have these nodes, these white ones are the nodes, and then you have edges that edges that is defined between 2 nodes. I'm going to generalize this. So you can have edges, hyper edges, not just between 2 nodes, but more than 2 nodes too. So let me try to explain this. What is it, what data I have here? So this is a toy data set I have here. Your vertices can be anything, and you're defining a relationship between these nodes. And what I'm also saying is that I do have this information. These edges could be weighted, or you have some ordering on these edges, and I give the color. I'm sure it's not very clear, but so there is a yellow color, green color, white color, and there are a bunch of other colors. And I'm saying that there is some ordering between them. So let's say yellow is less than green is less than white, or so on, less than or whatever. So there is some ordering, okay? And good question. The color is not on the vertices. The vertices are what they are. I know the color is white on the vertices, but ignore that color. I am only coloring the edges, okay? And I'm giving you an ordering on the coloring of these edges. The edges I'm showing you here is the regular edge between 2 nodes, but I had also showed you edges between 3 nodes, so I want to kind of, so that's an edge, that is a white edge between 3 nodes. So it's 2 simplex, as they call it. So that's a hyper edge. And these are the other edges that I had, one edge here, one edge here, and one more edge here. These are edges defined over 3 nodes, each of them. And there is no geometry here, but you can imagine that it is a tetrahedron, and that is a hollow tetrahedron. Why hollow? It would have been filled if I said that there is an edge between the 4 vertices. But I'm saying there is no edge between the 4 vertices in this example. So I can imagine that it is a tetrahedron and so on. So that's my data set. And that's the way it is defined. It so happens that I have this relationship between not just pairs. I always think people do only pairs because it is easy for us to understand, it is easy for us to visualize and so on. I think intrinsically we have this notion of properties across multiple, at the same level kind of a thing, multiple vertices and so on. And now my question is that, can I summarize what I show you on that little carpet, that brown background? I have this data set. Is there a way of summarizing? Of course, there's a way of summarizing, saying you have this many vertices, this many 1 simplex, this many 2 simplexes, and so on, something more. I'm saying something more sophisticated. Is there a way of summarizing this in a way that I can compare this summary with other such graphs that I can get? The simplicial complexes, let me just call them graphs, generalized graphs, for undirected, no direction, okay, this point, yeah, no direction. And I'm just asking a question is, what are the ways of getting some summary or some information from this that I can exploit in some algebraic form or compare and so on and so forth. So that's where TDA comes in, yes. I'm sorry? Yes. You can call it a plane. If you're comfortable with that, you call it a plane. But I'm going to treat it as an hyperedge because the treatment is going to be different in the TDA as we go. It's good for you to visualize it that way. But when I go to 4 edges, 5 edges, 6 edges, dimensions, not edges, dimension, I can go to 100 dimension. Then it becomes harder and harder to visualize, right? So yes, in the lower dimensions, we visualize that way. And these are just relationships, okay? The geometry I'm showing for us to visualize and appreciate whether when something is filled or not, that's why I was just showing, but that's for visualization. But at this point, there is no geometry in it, right? I didn't give you coordinates of that. I just said this is the relationship, but even with that relationship, I can get. So now topology comes in. So I have this data set. So I'm going to do TDA on this, which is topological data analysis. I will do it on this simple, small example, and then we'll. So because I showed you there was an ordering, I can say that I begin with the vertices that I had. So I have 11 vertices, and the number of connected components, that's Betty number 0, which is that. So let's say there are connected components, there is 11. Now I had an ordering of the edges, and I will introduce them in that order. So when I introduce first the yellow edges, because in my thing they were the lowest, I will have, of course, 11 vertices, 7 edges, and number of connected components now is 4. Okay, that is Betty number 0. And now I add the green edges, and this you can immediately count. You have number of connected components, and now there's something non-trivial that comes in, which is the Betty number 1. I'll not go into details of that, but what it says is that how many holes or how many cycles do I have, and the 1 stands for dimension 1. So I have 1 hole here, and I'm counting as 2 cycles. So this is a summary of the data. So this is, if I have this data, this is a kind of a summary. What you're seeing in white on the left, it's kind of obvious. You're just counting and saying. What you're seeing on the right is a little more sophisticated computation is required. At this point, we don't have to say what computation we did, but just to recognize that these are the characteristics we are looking at. And then I introduce the white. Then I introduce this 2 simplex now. So this is a 2 simplex. So I have 2 simplex 1. The edges can be called as a 1 simplex 11 edges, vertices. And now I have 4 connected components. And I have B1 as 3. Here I had B1 as 2, B1 as 3, because now this hole no longer counts because I filled it up. It's no longer a hole. But there are many holes here. I can get many cycles here. And if you count, you'll see there is actually more than 3 cycles. But why am I saying that there are only 3 cycles? And that's because this is a vector space, and we only count the, we take the size of the basis as this. And any other, the 4th cycle that you will get will be a linear combination of these 3. So that Betti number 1 is 3 here. There is a little bit of a problem when I start doing data analysis on this. I'll come back to that, because you probably may recall that basis is not unique. So which are the elements I'm talking about? But that's an issue, and we'll handle it later. And then I add my 2 simplices, the edges that I had shown you before in my diagram, and then you see that now it gets more complex. You have beta 1 is 0 because I lost all the first dimensional cycles, and I have beta 2 as 1. And why is that as 1 is because in this structure that I showed you here, it is a hollow tetrahedron, and that's the whole of a 2nd dimension. So beta 2 is 1. So that's why I had shown this example. Now you can imagine that in higher dimensions, I can have much higher dimensions, which I will not be able to visualize that easily that I can visualize here, but they exist. And so this is the summary. This is the summary that we have. Okay, so this is a summary that I could use. But since I had this ordering of edges, there is also this notion of bar diagrams that I can have at the bottom. Bar diagrams is actually tracking the birth and death of these holes. Like, for example, we saw that I had a hole here, right? But it died once I filled it with this one. Okay, so there's a natural birth and death, and that's captured in this bottom bar diagram. This is one way of visualizing the bar diagram. There are other ways of visualizing, but it is capturing some information, something about the data that I had. Once I have some ordering, and so 3 things to note here. So you have this simplicial complex. Simplicial complex simply means a generalized graph. Let's say there are different terms. You can have Uterius, Rips, check, and so on. It doesn't matter. There are different kinds of simplicial complexes you have. You have this notion of filtration of the simplicial complex, which is what I took you through these steps. I added these edges as I went. You're just adding. You're not subtracting. You just keep adding, and that's called the filtration. So there's a notion of filtration. And then finally, you have this bar diagram, which is a summary of that data set. I'm going to call it as a summary of the data set, or the property of my data that I got, which is tracking birth and death of homologies. And these are robust mathematical concepts, and they are robust concepts which you can actually compute and have. So these are the properties that we would have. So to summarize, that's the data set that I had, and I gave you the ordering of that. I can have a bar diagram that corresponds to that, and then I can utilize this. So this is just to summarize what I had just said. So we had this notion of course, we are using topology. We had this notion of 1, 0, 1, 2, 3 simplex, or in terms of higher edges. And then we have this notion of Betti numbers, which are holes of different dimensions. And then you have different kinds of complexes. And here is an example that I've taken from Grice's paper, which is a slightly non-trivial example compared to what I had shown you here. The one that I had developed was really small, but this is a slightly more complex example where a bar diagram would look something like this, okay, of different dimensions, and this is the filtration of this data, of that data set, which is actually a donut that he had taken and so on. So we have a tool called Matilda, and I will encourage you to take a look here and play with it, which can do these computations for you. The computations of the bar diagram and all these other things, these are slightly non-trivial computations. It can do all these, and you can use it as a library or sort of standalone applications, and so on. So now I want to use this. Okay, so I want to use what I said in a couple of examples that I want to show you. How do I use these things? So I'll give 2 examples. One is from population genomics. Yesterday we had a whole morning session was on population genomics. So I want to take one problem from there, and I'll talk about COVID also was covered yesterday. And here in phenomics, I will show you how we have used this. So, well, The population genomics, just to set it up, we were involved with National Geographic on a project called the Genographic Project, which had many PIs and so on. We are collecting data. That was probably 15 years ago. And that's where we had looked at some of these problems, and the problem inspiration comes from this. We had an out of Africa route. I'll not go into details of that. I think Professor Majumdar had covered yesterday a lot in this space. And he also covered the population model yesterday, except that his description and my description are flipped. You just have to flip it in your head. So let me just recap quickly what the population model is. Here, every line is a generation. Every little circle here is an individual. And this is the next generation. This is where I mean it is flipped, because your extant units of today are here, and this is past. So this is past and this is present. And the arrow actually shows the transmission of the genetic material, so your offsprings. And one difference is he was talking about the coalescent model yesterday. So here I will be talking about two parents. We're looking at admixture and so on. So we're looking at two parents. So they will. If I take 4 individuals here and try to get a common history, it is not a tree, it is a DAG. That's the difference from the coalescent model yesterday, which was a DAG. Here it's a tree, which is called ancestral recombinations graph. So because every node has 2 parents. And so we are looking at recombining nodes here. So this is the population model. And it's a panmictic population model, meaning that every node here, the parents of every node is picked, say, at random from that entire generation. Okay, so it's not grouped. So this is panmictic. And admixture in this setting, I will define as here was a population happily evolving like this, and here was another population evolving again like this, and at some point they came together. So this was panmictic across this, panmictic across this, but now this is panmictic across the two, and then they evolve. And so this I would say as admixture. Okay, and so it's kind of believed that there was some structure, some information here, and some information here, and there is some other information here as the population is evolving that you see from the individuals. You can reconstruct or capture that. So there is some pattern, something there that's going on, and that's through mutations and so on. So the question I'm going to ask is a very simple question, which is on the deep ancestry, or the kind of admixture that I showed you here. So if the problem is that if I give you k populations, each with multiple individuals, defined as a sequence of polymorphic sites or SNPs, okay, we are not interested in which are common SNPs, which are these populations that are admixed? I call them deeply admixed because you'll assume that you know the entire history, and that's the question you ask. So it's in a slightly different setting I'm asking the question. Of course, there are other approaches. Yesterday Professor Majumdar talked about those. There are indeed other approaches, but I am trying to pose a problem differently where we can utilize some other tool. So to understand this, these are 4 different scenarios. Okay, so I have 4 populations, A, B, C, D, and this is how in 4 different scenarios they have evolved. So let me just ask a question here. So given the 4 different scenarios, where is there admixture happening? In which of the scenarios are you seeing admixture? The whole is a good clue, yeah. So there is no admixture here. There is one admixture here, that of this, and there is still again only one admixture here, that of C. And there are 2 admixtures here of this. And the other way to see is that multiple paths, if I go to the root, then there is admixture. But that was a good observation, that when there is one big hole, there is one admixture, 2 big holes, there are 2 admixtures, and so on. Okay? So if I knew this entire history, this is how these holes will kind of relate to that. And we took inspiration from this paper on viral evolution by Rahul Rabadan and et al., where they looked at viral evolution, and when they saw a cycle, they said that there is some horizontal gene transfer as well and so on. So they were looking for the presence of holes in the phylogeny or in this common history structure that you could get. Our problem is a little different, a little more difficult, because if we take this ARG picture, there are holes everywhere because there is admixture. And when I zoom out, I see this structure, right? I see this big structure. So there are small holes everywhere, and I want to distinguish this big hole from, these are the very big holes, and I want to distinguish it from the small holes. So from there, if I can get a scaffolding out of this, I get these two big holes. And these big holes I want to distinguish from the small holes. Okay? And there's a theorem there which now connects it to the topology that I've said, that it does say that you can get holes from here that connect to the holes here and so on. We don't have to kind of get into that, but we get the essence of it, is that I can use the homologies that I get through TDA on this potentially. Because we also are, the problem is even more complicated because we have data only at this level. We don't have actually access to this structure. We are assuming that if such a structure existed, like the viral evolution in Rahul Rabadan's paper, that you take the extant units, you have their sequences. So there is a structure that is defining the relationship between these, and then you are looking at some holes in that structure. So it's not directly comparing, taking a small hole, big hole, and so on. You kind of have to, in abstract, they exist. And so this homology computation is going to pull them out. So that's the model that we use. I didn't go into the details of that. The details are there in the paper if you want to say what was involved. So we use this on simulator data, and the real data that we used is from plant genetics in avocado. We got it from our collaborators, plant geneticists, who knew the ones that were admixed and so on, and we were able to use our method using topology, using TDA to actually say which of the species, which of the samples were admixed and not. Yes? No, so you have to read the paper, Kamal. And that's why I said that you have these things only at the bottom. We don't have to reconstruct and then do the whole. Yeah. So there is no explicit reconstruction. You're just, yeah. And the second question The second question is that we just said any of these populations, are they deeply admixed? We said there's a big hole and that's admixed. The next question is, which are the populations? And there is a difficulty, I just want to say a mathematical difficulty here that of course we address, is that you start with data and you get the bar diagrams. And then you look at the bar diagrams and say, oh, there's an admixture, there's no admixture, and so on. But now if you ask the question, which population was admixed, you have to go back to the data. And going back to the data has a little bit of a mathematical hitch because, like I said, when the bars don't associate uniquely as in the the vanilla TDA uniquely to your data set because the basis is not unique. So you can assign them to different elements. So we had to do some mathematical development there, which introduced the idea of what are called essential simplices, and there have been other further developments we have developed on that with harmonic persistent homology and so on. These, in other words, are robust ways of uniquely mapping from a definition. We have a way of, mapping uniquely from the bars to the data, okay? And there is a definition of that. There are mathematical notions from that. If you use Matilda, it will kind of give you, if you want to understand the math behind it and you want to convince yourself that is what works for your application, you can look at the papers and so on. But this actually gives a way of uniquely going back. So I go back to the original problem there of simulations, and now if I want to say which are the populations that are admixed, I have to invoke this essential simplices and so on, and then we ran them on different simulated scenarios where we knew what the answers were, and we were able to see which were the populations. In fact, when we tried to use this on real data, the difficulty was that we were, human data and so on is very difficult to access. We have to get permission from that. We tried different sources. The genographic project was at a different level because the data that was collected. So we stayed with the simulation. We were happy with the fact that at least we know the model works in extremely complex scenarios like this and so on. So that is where we showed our results in the paper. So the second example that I wanted to see the use of TDA. So I thought that was a natural use of TDA, and it was good. Raul Rabadan had a paper which actually directly connected to the holes. You can always relate directly whether there's a hole or not. But we were able to take it to admixture and show that. So now I want to show you another example, different kind of use here. And the topic had logic and topology and so on, and Kamal was asking me, what is logic, what is topology, how are you using? So there are a lot of terms we are using. So I want to see how, I'll show you how we are loosely connecting all these things that we are saying. I showed you the connection between data and topology. That was through TDA, topological data analysis. And data and logic can be connected very simply through what is called redescriptions. I'll give a definition of that. It is very simple in terms of we are forming logical formulas from the data that you'd have. It's simple to the extent that you say, for example, if you say a sample has SNP1 and SNP2 and SNP3, we just say SNP1 and SNP2 and SNP3, and that's my kind of expression. And it could have been anything else as well. So that's how we'll get these logical expressions. We do also more complex things with that, but I will not be talking about them here. They're kind of work in progress, and I'm not going to be talking about that. For this talk, it is enough that we just understand it at that level. And then we can actually do topology on the redescriptions through persistence. Persistence is, I probably didn't use the word persistence before, the homologies that we are seeing. And persistence meaning that the homology stays for a long time. It persists for a long time. And that is a natural way of dealing with noise in this scenario in our model. So something that persists for a long time is possibly a signal and not noise. If something is born and dies early, I don't know why it is. Put it down? Oh, okay. Thanks. You have all the hacks, yeah. So the persistence is a way of actually dealing naturally in this space. So when you have this tool set of TDA, it is coming with getting your summary, but also a way of handling noise in this, yeah? Okay. So this is the connection that I have, the loose connection between logic, topology, and other things that is here. And the next example that I will show you will sort of use these. So when COVID, what did you say, broke out, arrived, or whatever, there were a lot of papers. And this is one of the papers that you may have seen because we knew so little about COVID and we were seeing this. And one of these remarkable things was that the patients were displaying multiple symptoms, right? Many symptoms here like anosmia, loss of smell, cough, fatigue, diarrhea, shortness of breath, fever. So there are kind of 6 symptoms that they were showing. And this was a very nice paper with this nice picture of a Venn diagram of how many, which was a combination of these symptoms. This is a Venn diagram of that. For example, if you were here, that means they saw 25 examples of, which is a dark one here, fever, and maybe diarrhea or something, I think, okay? So this is a Venn diagram. So my question was, when I looked at this, I said, yeah, it's a good summary of what has happened to all 500 or whatever patients. This many had this many had this. But can I build any hypothesis from this? Can I learn anything from this? Is this telling me something other than the Venn diagram itself. Okay, so that's the question we ask, and then we try to answer that. In terms of, can I get some hypothesis? So you'll go to hypothesis generation. So we use redescriptions, like I defined, I had said before. We'll take atomic logic variables from this data. We can trim it down. So we take this data and we work on it, okay? So of course, when we tried to contact the authors for these papers, it was difficult to get their data set which they had used, the process data, but we got to the raw data that had been there. So we are not getting the 6 symptoms, so we take about 1,500 patients, and then we take their entire electronic health records, the EHR, and get the coding from that, so that instead of getting just the symptoms, we are going to get some other descriptors for all these, for all the patients that we have. And we trimmed these down to, we trimmed this down to 31 features, okay? So these are the symptoms and the codes, this is 31. And now what will my Venn diagram now look like? Because the previous one was a Venn diagram on 6 sets. Now this is a Venn diagram on 31 sets. Well, this is actually, we didn't even try it. I just pulled out from the web a very complex Venn diagram, because this visualization doesn't help when you have so many, so there's none. Well, we can't visualize it, but we can analyze it, right? So we can use some tools on it to analyze. So that's the summary that you get from this data set. So you can just see that. So what it means here is that you have 651 patients who had exactly one of these symptoms or features, let's call it. And there were 431 which had 2. That means that some 2 of these 31, any 2 and so on. That's just a summary of that. But we want to kind of analyze further. So we use TDA, which is a good tool for that. And here this is something called a check complex. This is a cartoon figure of check complex. It doesn't represent the data per se. So we have 2 sets. So each set is a redescription. So each set you are having a combination of your symptoms or your features, and then you can have a notion of distance between 2 sets, and we use a Jacquard distance between the 2. So you can have, because these are individuals, and then when you have the filtration, you can connect these edges, or you get an edge between this set, which is, now this set is my vertex. What I had shown you in TDA, this is one node. Each set is a node, okay? That node gets fixed. And then there's a relationship that we do the filtration over the relationship. And that relationship is the distance that you have between these two, where you use a Jacquard distance. And the filtration you can do is, you can start the Jacquard distances from 0 to 1, you can start on small intervals going from 0 to 1, or the other way around, depending on how you set up your problem. And then you have an edge, one edge between 2, if the Jacquard distance between them is less than whatever threshold you specified, because that's the filtration you are doing. And between 3 you will have, when the Jacquard distance between all of them is less than 3. There is an extra thing that you have to say, that your problem setup actually leads you to simplicial complexes and so on, but we don't have, I'm not getting into that level of technical details. It works for this problem that we have here. And then we do the bar diagram for that of different dimensions, and then we pull out the bars, the long and lower bars, as potential hypothesis, okay? So in this one, for example, one of the hypotheses that we pulled out, is that some COVID patients had cough and some COVID patients did not have cough, independent of whether there was sputum or not. This is what we are getting from the data, and the hypothesis is that, do COVID patients with the cough and COVID patients without cough have different etiologies in the disease? Is the disease etiology different in the two? Is an hypothesis, because the hypothesis we get is that they should be different from the data that we are looking at. But to check this hypothesis, of course, it means that you have to do your experiments, and it's up to the clinicians or biologists to see what they would do. But we would pull out what we think are the hypotheses from here which has crossed this barrier in some sense of being significant and so on, and is getting from this data. So I showed you just one of these hypotheses, but this is an automated way of deriving the hypothesis using this sort of a tool. So we did a similar experiment with the UK Biobank data, where we take about 1,600 diagnosed COVID positives, out of which 1,000 were severe, and then for the features we take 16 labels and sort of define 150 redescriptions. It's like the nodes of the thing that we sort of fix, and this is a visualization of that. It doesn't matter, the visualization doesn't tell you much because you cannot make much out of it when the dimension is high or the data set is large and so on. But you can run your TDA and get the summary of these things like I was showing you there before. And so we do this, we pull out some barcodes at some dimensions, and the hypothesis that, again I'm going to show you one hypothesis, which is again interesting, is that diabetes leads to severity and obesity is a risk factor. But with respect to COVID severity, obesity and diabetes operate independently, is what we observed from the data set. That means that being obese doesn't make the impact of diabetes on COVID any more than without. It's a non-intuitive hypothesis, but this is what we are getting from the data. That means independently obesity and this one, diabetes and this, but the combination doesn't make it any worse as being severe COVID and so on. And in fact, there was another hypothesis that we had, which is that, so TDA links severe COVID tend to RAS and hyperlipidemia associated metabolic syndrome conditions, which is something that was even independently reported. I mean, this hypothesis was strong enough because we had seen that being reported in other papers, that we just had a whole paper on this single hypothesis, with this kind of title. So there were many other hypotheses. The point is that when you look at scaling discovery, when you want to discover at scale and so on, so this is a nice automated way of getting hypothesis. There are different, we are not saying it replaces anything else. This is a different way of looking at the data. You have the lens of TDA that you're looking at the data, and then you can generate hypotheses at scale. So to summarize, I showed you an example of population genomics where we used topology for getting topological signatures. So we got some signature, meaning some patterns of the bar diagrams, and we were concluding from that. I showed you another example from phenomics, COVID, where we try to combine redescriptions, logic, and topology, and so on to do hypothesis generation. So you can actually use this topology in principle, the TDA, in very creative ways of fitting to your problems and getting answers. It is not easy. I must say that it is not easy. It is it is difficult, but to me it is very interesting because it gives you a completely different way of looking at data. So then I'm going to just go back. So this is kind of the end of my technical part. I'm going to stop. Before I stop, I just want to go back to the beginning of it, which we said that the turn of the century, there is an explosion in compute and so on. And so one of the things that we are currently looking at is quantum computing, looking at quantum algorithms. So one of the things we are currently exploring is on cell-centric therapeutics using quantum algorithms. Is there an advantage? Can we get an advantage? And then we have a couple of papers in archive at this point, one on clinical trial design of optimization, looking at quantum algorithms there. And the first one that I said is most of the omics data processing. My interest and my close group's interest is kind of on using omics data. There are other applications of quantum computing that's happening. My focus is on, can we get something from omics data and using quantum. So let me finish by acknowledging, of course, the work that I showed has a lot of collaborators. The genographic project had many collaborators and so on. This is a working group. This is the most recent one, that the picture that you see on the left of the working group on quantum. And this is my, my team that is co-located with me in IBM TJ Watson in Yorktown. And thank you for your attention.
Questions. I'll prioritize the questions from students. Sorry. So, yeah, please. Sorry for that. So, yeah, I'll prioritize.
Hello, ma'am. Stop. I just want to know, can a hyperedge exist for the same vertices? Like, if we have vertices that is connecting to itself for multiple features, can an hyperedge exist for that?
An edge, a self-edge.
Yeah.
Good question. Self-edge is a self-edge. Your question is whether, how does it fit into the TDA here? Yeah. I'll have to get back on that. I'll also need to probably understand a little better what we mean by a, so the edge here, I'm saying in terms of relationship. Yeah. So when it has a self-relationship, what does it entail? What does it mean? So that one has to see whether the modeling is appropriate for the problems you might have in mind.
Okay.
Because we can probably hack our way through with the math on making it work and so on. Probably more importantly, if you have an application, we want to see that how does it relate, how does it correspond to that, and does it make sense?
Very nice talk. So yesterday we had a talk where it was discussed about Boolean networks and generability network setup. So is the TDA, can TDA assist in that?
It does. Very good question. The same thought was going through my mind when I was listening to Dr. Samal's talk. I did talk to him about it, and I'll tell you what my thoughts on that are, is that, okay, there were two thoughts, quantum computing and TDA. So TDA, of course, one has to see whether you can actually apply there, and again, like I said, does it make sense in the context? So Dr. Samal, for example, had many questions that he was asking, you know, about the networks that he had. And there'll be networks you'll be talking about later also. And if we model it this way, and the structures that we are getting, does it make sense to the questions that you are asking? And does it answer something in a unique way that would be different? Certainly worth exploring. Like I said, the connection is not obvious. Most of the problems, even the problems that I presented, when we began with, we didn't think there was any connection. You know, we kind of worked through it, and then we see that we can make the connection. So it is worth trying for networks, you know, because for graphs and all that, you have these natural filtrations that one defines. And then the question is, does it answer what you wanted? Or does it answer a question, interesting question that you had not posed, but you could pose and see that that's the answer you're getting?
And add on to that, just to be precise, so if you want to model the activation or inhibition as like connections between two nodes, how do you do that in the TDA framework?
So that's a 0-1 kind of the weight you can have, right? So you can probably think about the 0-1 and take them as weights like 0 and 1, right? And
thank you.
Sure.
So let's take that question and then we'll come in front.
Nice talk. I just wanted to ask a general question. So if you already know the nature of your graphical data, I mean the data, graph data, such as, for instance, say it's a scale-free network. So can you infer something in the TDS about its homologies? So what I'm trying to say is, if you have some incomplete information, can you skip steps to infer something about your data?
So the question you are saying is that you have incomplete information. Sorry, what is the what would you like to solve? Yeah.
About the graph. For instance, you already know that it has some high global clustering coefficient.
Could you, sorry, repeat that again? I couldn't hear.
What I'm trying to say is that if you already know the graph structure, then does it reflect in the homologies?
Yeah, so homology in a graph that you can actually see in the two-dimensional, if you have only say one simplex, cycles are the holes, right? So that will be the, that is the kind of the obvious structure that you will see. And the TDA is actually exploiting homology because it is based on sound math kind of a thing, and the homologies is being exploited. Now if your graph gets higher dimensional is when it gets more interesting, I would think. So that is why when you are in lower dimensions, you can actually have more efficient graph algorithms to solve your problems, because these are complicated computations that you are doing to infer homologies and so on. So if your graph is two-dimensional and so on, then I would say that one-dimensional graph algorithms would answer most of your questions.
Thank you.
First of all, congratulating for your excellent presentation. In one of your slides, you have shown that quantum computing on cell therapy, right? So, right, I am asking about how DNA computing goes on in cell therapy as such.
Sorry, how?
DNA computing goes hand in hand for cell therapy as such.
Okay, so that was on the last couple of slides.
Yes.
So DNA computing there, well, we'll have not, you mean genomes and so on, right?
Yes. Yes,
so this is actually, you will take the data, just like you would do machine learning, say, on omics data and so on. You have your data, expression data, or you have your sequence data or anything, and just like you would say, do machine learning, statistical learning on that. So you can think of quantum computing in some sense. You have encoded your data in some form. There can be other ways, encoded data in some form, and you're trying to use machine learning type of things, say, with your quantum computer. So you'll have some quantum algorithms that are working on this. This is ongoing work. This is all very new work, right? So, yeah. I mean, you can come up with even more creative ways of representing your data or encoding your data so that it is exploited well by quantum computing or the quantum information computing models.
Thank you so much.
Can we have?
So on the first part of your talk on admixture, so I've been following the literature on these phylogenetic networks for a long time, and it seems surprising that you're able to find these deep admixtures, because I could imagine a process which generates holes of all scales, small ones and bubbles of various sizes. So how are you able to, is there an assumption of a separation of scales between the small recombinations and the large admixtures?
Yes, yes. So of course there's a thresholding that we'll say. So roughly speaking, what you're saying is that, can I get long bars? If I get very long bars, it persisted and it is a large hole. So it is a thresholding thing, and you say that beyond that hole, beyond that threshold, if I have a bar, it is as long as this, then possibly it is, it's a hole from, that bigger picture. Yes.
Just a random question, which is, you have this tree. Can you say something about the rate of growth of the tree? Meaning, the question I'm asking is the growth of the population, can it be depicted in some way, or can it be analyzed?
Right. So these are, if you're talking about the growth and other things, that was not what we were modeling, right? But you can use other methods to solve that problem. But if you want topology to actually address that, then we will have to study it, put it under simulated conditions and see, because because it won't be a mathematical proof in that sense. It will be just like I showed you. It was, we had a proof of certain conjectures we had to begin with, saying can we connect small holes with big holes. So we had some mathematical proof on that. Yes, we can. And then the next is, can we actually get it from data? So that we had to do under simulated conditions. So we simulated data, produced some extant sequences, and then applied this topology and so on. So there's a Vietoris-Rips complex and other things that we are using in that and so on. So if you have a different question, then and we want to use topology, then we have to sit down and see how we can do it. But otherwise, this question can also maybe tackled by other means. There are a lot of other statistical and other methods that exist.
We can take 2 more questions.
Yes, so theoretically, everything looks all right, but when you're doing practical experiments, we are trying to use some of these kinds of analysis. You find duplicate nodes, existing in the data. So how to disambiguate?
So what data are you talking about?
So we are using this for NMR, analysis, trying to deconvolve the spectra and come up with some patterns that can tell us about the molecule itself. So some parts, work fine. When we do synthetic, based on synthetic data, because the experiments sometimes are not well resolved, so that the peaks are separated out clearly, like you can see that they are different. Sometimes they're overlapped. So for cases where it's clear, this works perfectly fine, in fact.
You're saying TDA works there?
Yes, it works perfectly fine, but sometimes the resolution is not good, so there are overlaps, in which creates a graph where you have duplicate nodes. They're sitting right there.
Right.
So what are the ideas that one can use to kind of solve such problems when you have duplicate nodes?
Right, so that's a class of problems that I have not spent time on, where where you can model your questions or model your data in actual 3D space, and then you're running topology on that. And I will have to probably take a look at it closely. But I think there are a lot of other works also in that space. I do not know if in literature they have kind of addressed these questions already or not. I don't know, but I don't have an
these
answer.
Kinds of questions have been addressed by other techniques.
Other techniques.
Yeah, other techniques, yes.
Yeah, I don't have an answer from the top of my head, but probably have to think through it. For instance, yeah, we'll have to look at whether it is an interesting question to ask, and whether it actually poses even sort of basic mathematical questions, and there is a mathematical solution, just like we did for the essential simplices, with the harmonics and so on. So whether it can be studied at that level and introduce, it can be an interesting exercise. Yeah.
Thank you.
So I have one question regarding the use of Matilda, so which you showed there. So can we see the relatedness of 2 transcriptomics data? I mean, transcriptomic data coming from 2 different systems. Can they be, interlinked via that sort of using Matilda?
Yes. We hope so. Like I said, we actually have played quite a bit with it, with many kinds of transcriptomic and other data sets. But happy to talk to you and to see what specific questions you have in mind and whether we can model. So let me also say that we did play around with it and get bar diagrams from transcriptomic data in different settings, but we were not able to take it to some interesting point of answering some interesting questions and so on. It doesn't mean that the specific question that you have in mind we cannot, we can't solve or we can get there.
As I saw in your COVID work, you found the relatedness of certain specific features. These are related. So that is the sort of thing which maybe we can talk over later.
Oh, absolutely,
so,
absolutely.
Maybe transcriptomic data of 2 systems, we want to see the relatedness between them.
Yes.
Because we have found a link, And that is why we wanted to see that how far they are related. So maybe this process might
Yeah,
work better.
totally. So let's talk about it. And,
sure.
yeah.
Sure, thank you.
And we have other tools also that can help. So other basic tools, we can bring them in.
Okay, let's thank Dr. Laxmi again. So for more questions, please use the coffee break now. So we'll break for coffee until 10:30. And then the next session on network biology will start at 10:30.